So now we are going to see about the endomembrane system. So generally what is endomembrane system we are going to see. So here endomembrane system is nothing but some of the organelles, membrane bound organelles. So they are coordinated to perform certain functions.
Okay.
So this system, those organelles which are coordinated, those organelles together called as, so those organelles together called as endomembrane system.
Endomembrane system.
Yes.
Okay.
So in many membrane bound organelles perform functions which are coordinated. Okay. So those organelles together are known as endomembrane system.
Okay.
So because of these are membrane bound. So what are the organelles involved here? So these are endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. Okay, so these are coordinated and does functions and do their coordinated functions. So if you see examples like mitochondria, chloroplast and peroxisomes, these are not coordinated. They do functions, but these are not coordinated from one organelle to other organelle. So therefore these are known as non-endomembrane system. These do not do any function, so these does not come under endomembrane system. Since these functions are coordinated and linked from one organelle to another organelle, it is known as endomembrane system. So endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. So first we will see about endoplasmic reticulum. Okay. So these endoplasmic reticulum arise, they are attached nearby to the nucleus.
Okay.
So they are tubular and they form a network of reticulum-like structures like that. Okay, so these are often found near the nucleus and near the nuclear pore and everything. So here when ribosomes are attached to this endoplasmic reticulum, it is known as rough endoplasmic reticulum. When ribosomes are attached to it and they are rough. So if these endoplasmic reticulum do not have ribosomes on them, and these when these appear smooth, these are known as smooth endoplasmic reticulum.
Okay.
So generally rough endoplasmic reticulum synthesizes protein. It involves in protein synthesis and secretion. Okay, so it synthesizes protein, and they are extremely continuous. They are continuous with the outer membrane of the outer membrane of the nucleus. Okay. So here smooth endoplasmic is involved in the synthesis of lipids. Okay, it synthesizes lipids. In animal cells, lipid-like steroidal hormones are all synthesized by smooth endoplasmic reticulum. Okay. So this is about endoplasmic reticulum, which is the first subdivision under endomembrane system, which is the first category under endomembrane system. So next we are going to see the coordinated organelle which is Golgi complex. So how it is coordinated with endoplasmic reticulum, we are going to see that one. Next we will see about Golgi complex. Okay. So after endoplasmic reticulum, now we are going to see about Golgi complex, which is also known as Golgi apparatus. Okay. So it was first observed by Camillo Golgi. Okay.
So that it's named as Golgi Apparatus.
Yes. So he observed some densely stained structures, reticular structures near this endoplasmic reticulum and near nucleus. Okay. So these were later named because of him, by him. So therefore it is known as Golgi apparatus. Okay. And these consists of cisternae, sac-like structures called cisternae, and with a diameter 0.5 to 1 micrometer. So it has two phases. First one is convex phase, which is the forming phase, and concave trans, which is maturing phase. Okay. So cis is for forming phase, trans is maturing phase. Okay. It is also known as convex cis, concave trans. Okay. So here the substances first move into the forming phase, and then it matures and it reaches maturing phase, and then it is released out. Okay. So here these Golgi apparatus do, these pack substances. So whatever the endoplasmic reticulum release substances, right, proteins and lipids, it is released, smooth endoplasmic and rough endoplasmic. So these materials are packed in Golgi apparatus. These materials are taken and they are packed like vesicles and they are transformed, okay, to the cytoplasm of the cell. So since they get materials from endoplasmic reticulum like proteins and lipids, they are found in close association with endoplasmic reticulum, okay. So here proteins are modified in cisternae inside this the sacs called cisternae, okay. So first what these proteins do, they first enter to forming phase and then enter to maturing phase and then they are released out, okay. So when proteins are packed, it is known as glycoproteins, and when lipids are packed, it is known as glycolipids. So this is the function of Golgi apparatus, okay. So endoplasmic reticulum synthesizes proteins and lipids, where Golgi apparatus, it packs the proteins and lipids. So it is both the functions are interconnected here, okay. So next we'll discuss about lysosomes, what it does and everything. Endomembrane system, we are after Golgi apparatus and the endoplasmic reticulum, we are going to see about lysosomes. So these lysosomes are membrane bound vesicular structures which are also known as lysosomal vesicles. So these are when the Golgi apparatus releases glycoproteins and glycolipids, these are transformed to lysosomal vesicles, okay. So these lysosomal vesicles, these isolated lysosomal vesicles are rich in hydrolytic enzymes, okay, like lipases, proteases, carbohydrases and nucleases and etc. So lipases generally digest lipids. Proteases for proteins, it digests proteins, and carbohydrates. Yes, and nucleases, it digests nucleic acids, okay. So all these hydrolytic enzymes does their own function, and these are active at acidic pH, okay. They are active at acidic pH which is below 7, which is below neutral. And here this is about lysosomes, okay. So next we are going to the fourth organelle in endomembrane system, which is vacuoles. So here vacuoles are nothing but generally which are large in plants and it's small in animal cells. So these, what are present inside vacuoles? So these contain water sap, excretory products and other materials not useful for the cell. So this vacuoles, this organelle has a membrane known as tonoplast. This tonoplast, what it does means it allows all the substances inside it. So it is freely it ensures the free movement of all ions and molecules and everything. So here therefore it is in plant cells vacuoles occupy 90% of the volume. So these are too large in plants. And if you see organisms like amoeba, there are some structures called contractile vacuoles, okay. Here this contractile vacuoles help amoeba to excrete, so it performs a role in excretion in amoeba, okay. And then if you see in protists, there are some food vacuoles, okay. What these protists do, they just engulf food particles and make them as food vacuoles inside them, okay. So this is generally about vacuoles and lysosomes, okay. So with this the endomembrane system is over. So next we are going to the next subtopic, which is mitochondria, okay. So now we are going to see the fourth subtopic under eukaryotic cells, which is mitochondria. So here this mitochondria are not easily visible. If they are stained by some chemicals, by some compounds, it can be visible, okay. So this mitochondria varies in cell. The number of mitochondria varies in a cell, and the length and the size of the mitochondria also varies in a cell according to the functions of the organism. The size and the shape varies in each organism. So diameter ranges from 0.2 to 1 micrometer. And its length varies from 1 to 4.1 micrometer, okay. So here this mitochondria consists of this mitochondria is double membrane bound structure, okay. It has two membranes, outer membrane and inner membrane. So outer membrane is generally continuous. They do not have any foldings or anything. They are continuous. Whereas inner membrane have a numerous infoldings, numerous structures infoldings. They increases the surface area of mitochondria. So these infoldings are known as cristae, like this. Okay, so if this is the outer membrane and this one is the inner membrane, right? So this one is the outer membrane and this one is the inner membrane, and this one is intermembranal space, and this one is matrix which is present inside the inner membrane. Okay, so it has outer compartment and inner compartment. Outer compartment and inner compartment both are aqueous. Okay, so here generally the site of aerobic respiration takes place in mitochondria. Mitochondria is the site of aerobic respiration for cellular energy, and so it produces ATP, adenosine triphosphate. We have already seen energy. Many organisms use ATP, adenosine triphosphate. So here cellular energy is produced here, ATP is produced here. So therefore these mitochondria are known as powerhouses of cell. Okay, powerhouses of cell, these mitochondria are known as. And so these mitochondria have in their matrix they will be having single circular DNA. So they will be having a single circular DNA, a few RNA molecules like 70S with 70S ribosomes, RNA molecules, and some components required for the synthesis of proteins. Okay, so these also synthesize proteins with those components. Okay, and these mitochondria divide by fission. Okay, they through fission they divide themselves, and this is about mitochondria. And each membrane, the outer compartment have their own specific enzymes and inner compartment have their own specific enzymes. It may be different or it may be same. Okay, so this is about mitochondria. The important thing is to remember here is it has two membranes, double membrane system, cisternae, very important, and cristae, which is the numerous infoldings of the inner membrane, cellular energy ATP, therefore it is known as powerhouses of cell. So these are the important points to remember in mitochondria. Okay, so next we will go to the fifth subtopic under eukaryotic cell, which is plastids. Okay, now we are going to see the fifth subtopic under eukaryotic cells, which are plastids. Okay, so here these plastids are present in all plant cells and euglenoids. So here these plastids are big in size, they are large in size, so therefore we can see them easily. Okay, so these plastids bear pigments in them. So based upon pigments, what kind of pigment it has, these plastids are divided into three types. Okay, chloroplasts, chromoplasts and leucoplasts.
Okay.
So here chloroplasts contain pigments like chlorophyll, carotenoid pigments. Okay, so with these pigments the cells perform photosynthesis. Okay, so this is about chloroplasts. We will see in detail about chloroplasts. Okay, so next chromoplasts. So these chromoplasts are fat soluble substances. It has pigment like carotenoid pigments, and the pigments are the carotenoid pigments like carotene, xanthophylls and etc. Okay, so these cells will contain carotene pigment and xanthophylл pigment, which are altogether known as carotenoid pigment. So this gives its color like yellow, orange or red in color. Okay, so then third kind is leucoplasts. So leucoplasts are known as colorless plastids. They do not have color. So these plastids are again divided into three. Leucoplasts are again divided into three: amyloplasts, elaioplasts and alluroplasts. Okay, so amyloplasts, yeah, it stores carbohydrates.
Example, potato.
Yes, and elaioplasts store oils and fats.
Okay.
Alluroplasts store proteins in them. Okay, so these are the three divisions under leucoplasts.
So alluroplast is an example of cashew nuts, can we say?
store proteins in them. Yeah, it is present in all generally cells. Okay, inside the cells these plastids are present in plant cells. Okay. So next we'll discuss in detail about chloroplasts, which are very important for photosynthesis. So in plastids, now we're going in detail about chloroplasts.
Chloroplast. Okay.
So already we have seen the chloroplast, chromoplast and leucoplast. In that we are going in detail about chloroplasts. Okay. So here chloroplasts generally are present in mesophyll cells of leaves.
Okay.
So because these chloroplasts contain pigments, chlorophyll pigments and carotenoids and everything.
So only they are present in the leaves.
Yes.
Okay.
Mainly they are present in leaves.
Okay.
So these help in photosynthesis and these produce food. And these vary in shape. They can be lens shaped or oval shaped, discoid or even ribbon-like. Yes, ribbon-like also they can be. And the length of these organelles are 5 to 10 millimeter and width up to 2 to 4 millimeter.
Millimeters.
Yes.
So it's a big.
Yes,
compared to
it's
other.
big. Okay. So these chloroplasts can be present one in cell in chlamydomonas and 20 to 40 in cell in green algae, which are present in mesophyll cells.
So like this 20 to 40.
Yeah, 20 to 40 chloroplasts separate. Okay. So it can be 20 to 40% in green algae. It varies in number. And this is also double membrane bounded organelle, like we saw in mitochondria. That it was also double membrane. So this chloroplast, it is also double membrane bound layer. Okay. So here the inner chloroplast membrane is less permeable to substances than the outer chloroplast membrane, outer membrane. Okay. So inner membrane is less permeable. So the space which is enclosed by this inner membrane now, it is called as stroma. Okay. So this one is called stroma. Okay. And these have a pile-like structure called thylakoids. These together known as thylakoids.
So these are group like such as the
Yes.
compartments.
Yeah.
Okay.
These are known as thylakoids. And grana are nothing but which form these thylakoids. Each one is known as grana. Okay.
Plural is thylakoids.
Not plural. The composition, if many granas join, it becomes thylakoids. Okay.
If a group joins as thylakoids, if it's single, it's grana.
Yes. Okay. So it is made up of coins like grana substances, grana. And there are flat membranes present in these chloroplasts named as stroma lamellae. So this stroma lamellae are nothing but this one. The connecting links between one thylakoid and another thylakoid is known as stroma lamellae.
Tube-like structure.
Yes. Okay. And these stroma of chloroplasts contain enzymes. The stroma is the inner cavity which is enclosed by the inner membrane. So these have a number of enzymes. Enzymes which are required
to synthesis
carbohydrates and proteins. Okay. So inside these stroma there are number of small double-stranded circular DNA molecules and the ribosomes are also present, as we saw in earlier. Chloroplasts and mitochondria have ribosomes within it. And chlorophyll, this pigment chlorophyll is present mainly in thylakoids.
Okay.
This is made up of grana.
Okay.
And here, what kind of ribosomes are present are 70S ribosomes are present here.
70S.
Yes. Okay. So which is made up of 50S less 30S. This is
70. Yeah, and last time we saw
yeah. Okay, so these kind of ribosomes are present here. And so this is about chloroplasts. Okay, so next we will see about ribosomes and cytoskeleton in eukaryotic cells. What are the structure of them?